Plants are made up of cells. Cells together form tissue, which joins to form an organ. The organs of plants include vegetative parts such as roots, stems and leaves, and reproductive parts such as flowers, fruits and seeds. The growth of plants takes place in two stages, namely vegetative and reproductive. The vegetative stage involves the growth of the plant from a seed to the flowering stage, whereas the reproductive stage is the period from flowering till they produce offspring. During the vegetative stage, the organs of the plant grow in size and number and result in a mature plant with complex body organization. This development of a mature plant involves two processes, growth and differentiation. Now, let's define growth. Growth is an important characteristic that is easily observed in all living organisms. It's a permanent increase in the size of an individual cell in a plant. It requires energy and hence accompanies anabolism and catabolism. In plants, growth is unlimited and hence they retain the capacity to grow throughout their lifespan. This is attributed to the presence of meristems at specific locations in them. Meristems are cells that have the capacity to divide, which add new cells to the plant body. This addition of new cells by meristems is called open form of growth. Meristems are of two types, namely apical and lateral meristem. Apical meristems are located at the root tips and shoot tips of all plants. They are responsible for the primary growth of the plant, that is, elongation of the plant along its axis. Lateral meristems such as vascular cambium and cork cambium are present in woody plants. They are responsible for the secondary growth of the plant, which causes an increase in the girth of plant parts such as the stem and branches. Now, let's see if we can measure growth. At the cellular level, growth refers to an increase in the amount of protoplasm, which is difficult to measure. Hence, growth is measured by different parameters such as size, area, length, and number. For example, watermelon cells increase up to 350,000 times, and hence the growth here is measured in size. In the case of Vittoria regia, a leaf, growth is measured as an increase in surface area, whereas the growth of the pollen tube is measured in length. Similarly, the root apical meristem of maize results in 17,500 new cells per hour, and hence growth here is measured in number. Now, let's look at the different phases of growth. The different phases of growth are zone of division, elongation, and maturation. The zone of division contains the rapidly dividing cells of meristems. These cells are characterized by rich protoplasm, large nuclei, and a thin primary cell wall with more plasmodesmatal openings. The zone of elongation lies next to meristematic tissues. These cells are characterized by enlarged cells, deposition of a new cell wall, a thin cytoplasmic layer, a large vacuole, and a nucleus. Maturation growth is seen on the cells next to elongation tissues. These cells are characterized by mature cells that have reached maximum size in terms of cell wall thickening and protoplasm. We know that a plant's growth comprises cell division, cell enlargement, and cell differentiation. When growth is measured per unit time, it is called growth rate. If we consider the increase in cell number as growth, it can be the result of arithmetic or geometric growth. Arithmetic growth refers to the division of one daughter cell obtained during mitosis, while the other daughter cell enlarges and differentiates. An example of arithmetic growth is the elongation of the root. Here, let's plot a graph to determine the growth rate of a root. Let's take the length of the root on the y-axis and the time taken for growth on the x-axis and plot a graph. A linear curve is obtained for arithmetic growth. This is due to the continuous addition of new cells at the root tips. Arithmetic growth can be expressed mathematically as LT equals L0 plus RT, where LT equals length of the root at time t, L0 equals length of the root at time 0, and RT equals A upon B, which is elongation of the root per unit time or growth rate. Geometric growth, on the other hand, refers to the division of both daughter cells obtained during mitosis. This type of growth is seen at the early stage of plant development, that is, development of the zygote to embryo. We can plot a graph for geometric growth by taking the length of the plant part on the y-axis and the time taken for growth on the x-axis. An S-shaped sigmoidal curve is obtained. The curve depicts the different phases of growth in living organisms under natural conditions. Growth is slower in the initial phase and is called the lag phase. Later, growth is rapid at an exponential rate and is called the elongation or log phase. Finally, growth slows and it is called the stationary phase. Geometric growth can also be expressed mathematically as W1 equals W0 e raised to RT, where W1 equals final size of the root, W0 equals initial size of the root at the beginning of the period, R equals relative growth rate, T equals time of growth, and E equals base of natural logarithms. The growth rate obtained here is called the relative growth rate. It is also called efficiency index as it measures the capacity of a plant part to produce new cells. Thus, we can see geometric growth during the development of the zygote to embryo and arithmetic growth in the later stages of development of the seed into a mature plant. Growth in plants can be quantitatively compared using absolute growth rate and relative growth rate. Absolute growth rate refers to the total growth of a plant part per unit time, while relative growth rate refers to the growth of a plant part per unit initial parameter. Let's compare the growth rate of 2 leaves. Here we can observe a 2 cm increase in the surface area of 2 leaves. This shows that the absolute growth rate, or AGR, is same for both the leaves. But the relative growth rate of the first leaf is greater compared to the second one. The difference in the relative growth rate may be due to the difference in growth conditions. Plants require specific conditions for growth. These include water, oxygen, nutrients, temperature, light, and gravity. The increase in cell size is due to the absorption of water. Water also acts as a medium for metabolic reactions required for growth. Oxygen is required by plants for the release of energy in food. Macro and micronutrients are used as energy sources and in the synthesis of cellular contents like protoplasm. In addition, plants need an optimum temperature for proper growth. The growth of plants depends on light, as they manufacture food by photosynthesis. Gravity determines the direction of shoot and root growth in plants. Plant development includes all stages of plant life, from germination of the seed to aging or senescence. In plants, development includes both an increase in the number of cells as well as cell differentiation. We know that root merostem, cambium, and shoot merostem add new cells. These new cells undergo structural changes for specific functions. This phenomenon is known as differentiation. For example, some of the meristematic cells of the procambium lose protoplasm and develop strong, elastic secondary cell walls to form vascular tissues. These tissues then help in the transportation of water and food in plants. In addition to vascular tissues, the procambium also differentiates into parenchymatous cells, which act as supporting tissues. Under certain conditions, some plant cells have the ability to regain the capacity of division after differentiation. This phenomenon is called de-differentiation. Differentiated parenchymatous cells de-differentiate to form meristematic cells, that is, the interfascicular cambium. Meristems formed by de-differentiation give rise to cells that lose the ability to divide but mature to perform specific functions, which is called redifferentiation. For example, the meristematic cells of the interfascicular cambium redifferentiate into the cortex. Differentiation in plants is open similar to growth. because cells formed from the same meristem differentiate into different structures at maturity. The final structure of a cell at maturity is determined by its position in the plant. For example, cells distal to root apical meristems differentiate into root cap cells, and cells at the periphery differentiate into the epidermis. Plants also develop different structures in response to different phases of life or the environment. This phenomenon is called plasticity. Heterophyly, the presence of more than one type of leaf, in the coriander plant is an example of plasticity in response to different phases of life. As you can see, the leaves of juvenile and mature coriander plants differ in shape. In the same way, the different shapes of the buttercup leaf above and below water are examples of plasticity in response to different environments. In the life cycle of plants, cell growth, cell differentiation, and cell development are interrelated and controlled by intrinsic and extrinsic factors. Intrinsic factors can be intracellular, such as genes, or intercellular, such as growth-regulating chemicals. Extrinsic factors include light, temperature, and the availability of water, oxygen, and nutrition. In plants, growth regulators or PGRs intrinsically control development. These regulators are chemical compounds and are also called plant hormones or phytochromes. PGRs include auxin, gibberellin, cytokinin, abscisic acid, and ethylene. They are divided into two groups: plant growth promoters and plant growth inhibitors. Plant growth promoters enhance growth activities like cell division, cell enlargement, flowering, fruiting, and seed formation. Auxin, gibberellin, and cytokinin act as plant growth promoters. Plant growth inhibitors respond to wounds and stress from biotic and/or abiotic factors by inhibiting growth. Abscission, the falling of leaves due to scars, and dormancy, or the quiet period of the plant during winter, are due to the action of plant growth inhibitors. Abscisic acid acts as a plant growth inhibitor, while ethylene can act both as a growth promoter and inhibitor. The discovery of PGRs was accidental. The discovery of auxin was initiated when Charles Darwin and his son Francis Darwin observed the coleoptile or canary grass exhibiting phototropism towards blue light. After several experiments, they found that the tip of the coleoptile is responsible for the bending of the full coleoptile. Later, F.W. Wendt discovered that a substance at the tip of the plant was responsible for its growth and called it auxin, which means to grow in Greek. But the chemical structure of auxin was identified when it was isolated from the urine of a patient by Kogel and Timan. The discovery of gibberellin is linked to E. Kurosawa, who observed abnormal tallness in rice seedlings struck by bacane or foolish seedling disease. This disease is caused by the fungus Gibberella fujikuroi. Later, Yabuta and Sumiki isolated the substance secreted by the fungus and called it gibberellin. The discovery of cytokinin is linked to F. Scooge and his co-workers, who noticed that cells extracted from the internodal segment of the tobacco stem proliferated to callus. When the nutrient medium is supplemented with extracts of vascular tissues, yeast and coconut milk, in addition to auxin. Later, it was Skoog and Miller who isolated and crystallized the substance that promoted cytokinesis in plant cells. It was then named kinetin. The discovery of abscisic acid dates back to the 1960s, when 3 independent researchers purified 3 different inhibitors of plant growth, namely inhibitor B, abscission 2, and dormin. Later, it was found that all 3 inhibitors were chemically identical and were called abscisic acid. The discovery of ethylene is credited to a scientist named Cousins, who observed the release of a volatile substance from ripened oranges. He also found that the volatile substance promoted ripening of other unripened oranges. Later, this volatile substance was identified as ethylene. Plant growth regulators or PGRs include auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Auxin is the generic name for all natural and synthetic compounds with definite growth-regulating features. Naturally occurring auxins include indole acetic acid or IAA and indole butyric acid or IBA, produced at the apex of plant stems and roots. Synthetic auxins include naphthalene acetic acid or NAA and 2,4-dichlorophenoxyacetic or 2,4-D. Auxins are used in plant propagation as they cause rooting in stem cuttings. They also induce flowering, as in pineapples, prevent falling of young leaves and fruit, and promote abscission of mature leaves and fruit. Auxins are used in parthenocarpy, that is, the formation of seedless fruit such as seedless tomatoes from unfertilized flowers. They exhibit apical dominance in higher plants, that is, they inhibit the growth of lateral buds. This is why the shoot tips of tea plants are cut or decapitated to initiate the growth of lateral buds. Synthetic auxin 2,4-D is used as a herbicide. It uses the physical difference between the leaves of monocots and dicots to selectively kill dicots. Hence, it is widely used in agriculture and horticulture. Gibberellins or gibberellic acids or GA are extracted from fungi and higher plants. They are referred to as GA1, GA2, GA3, etc., in the order of discovery, and they are acidic in nature. GA3 was the first of all the gibberellins to be structurally discovered. Gibberellins have varied physiological effects on plants, such as an increase in the length of the plant body axis. This is why they are used in the lengthening of grape stalks and sugarcane stems to increase the yield. Gibberellins also improve the fruit's shape by causing elongation as in apples. They delay senescence or aging of fruit, and are hence used commercially to extend the market period by retaining fruit on trees longer. However, they also hasten the maturity of seeds, and hence are used to obtain early seed production from juvenile conifers. Commercially, GA3 is also used in the brewing industry to increase the enzymatic activity inside grains during the malting process to achieve even germination in all seeds in commercial beer production. In cabbage, gibberellins promote bolting, that is, lengthening of internodes before flowering. Growth promoter cytokinin plays an important role in the cytokinesis or the division of cytoplasm in a plant cell. They were first discovered in the form of kinetin, a purine derivative. One type of cytokinin, zeatin, was later isolated from corn kernel. And much later, naturally occurring cytokinins were identified in specific regions of plants such as the root apex, shoot apex, and fruit to promote growth. They promote the growth of lateral and adventitious shoots by preventing apical dominance. They also delay senescence or the aging of leaves. Another growth promoter, ethylene, is present in plants in gaseous form. It is synthesized in the organs of plants that undergo senescence, such as ripened fruit. Commercially, it is available in aqueous form, ethaphon, which can be easily absorbed by plants. Ethylene gas is released from the absorbed ethaphon, which accelerates the ripening of fruit by enhancing the rate of respiration and is called respiratory climactic. This property is used in the artificial ripening of tomatoes and apples. It promotes the abscission of leaves and flowers, and thus favors thinning of cotton, walnut, and cherry. It also promotes the apical hook formation in dicot seeds to protect the apical part of the shoot that emerges during germination. It breaks seed and bud dormancy and is hence used to enhance the germination of peanut seeds and potato tubers. It also aids in the lengthening of internodes in rice plants, thus protecting the shoot from getting submerged in water. It also promotes root and root hair formation, thus helping the plants increase their absorption surface. It initiates flowering in some plants such as mango, pineapple, and cucumber, thus increasing their yield. Abscisic acid is a plant growth inhibitor that regulates abscission and dormancy in plants. It helps plants cope with stress and is hence known as a stress hormone. In the case of unfavorable environmental conditions, it inhibits germination of seeds. In the case of drought conditions, it closes the stomata, thus helping the plant withstand the stress caused by lack of water. In fact, the function of plant growth regulator abscisic acid is opposite to that of gibberellins. Plant growth regulators or PGRs control the growth and development of plants. In addition to PGRs, extrinsic factors such as light and temperature also influence the growth and development of plants. In fact, plants can perceive the duration of light and dark exposure and respond accordingly. For example, flowering in some plants is induced by their relative exposure to light and dark periods. This response of plants to the relative period of day or night is called photoperiodism. The site of perception of photoperiod is the leaves. Therefore, scientists hypothesized that these plants synthesize a flower-inducing hormone called florigen in their leaves. When these plants are subject to photoperiod, this hormone travels from the leaves to the shoot tips to induce flowering. Later, they found the PGRs to be the actual hormones that control flowering in plants. Based on their requirement of light, plants are categorized into long day, short day, and day-neutral plants. Long-day plants like strawberry and soybean need to be exposed to more than 12 hours of light to induce flowering. Short-day plants like pea and carrot need less than 12 hours of light exposure to induce flowering. While day-neutral plants such as sunflower and cucumber can produce flowering with more as well as less light exposure. Apart from light, temperature also influences flowering in some plants. For example, some plants produce more flowers when exposed to a specific period of low temperature. This kind of development of flowers due to the influence of low temperature is called vernalization. Since these plants need to be exposed to low temperatures for a specific period, vernalization provides the time for vegetative growth and hence prevents early flowering. Plants that exhibit vernalization include winter annuals and winter biennials. Winter annuals such as rice, wheat and barley are planted during autumn. Their vegetative growth starts in winter when they undergo vernalization, which induces them to flower during spring. Finally, they are harvested in summer. On the other hand, cabbage and carrot are winter biennials. Their vegetative growth starts in winter when they undergo vernalization, which induces them to flower during their second year. Thus, vernalization and photoperiod play a vital role in plant growth and development.